All right, I can start the second session of the workshop, day 2. Okay, so the outline of my speak is as follows. The first topic is how to utilize the spatial information. And the second one is the unmet needs in spatial transcriptomics. And the third one is interactive visualization tools in spatial biology. So there are a number of approaches to utilize the spatial information. When it comes to spatial transcriptomics, we should always think of what is the difference between the spatial omics data against the single cell omics data. And there are a number of approaches to deal with spatial information to make better research or to make novel research. And the first example is HMRF. HMRF is a variation of hidden marker model, HMM. HMM refers to a set of initial probabilities, state transition probabilities, and emission probabilities. And when HMM is considered in a spatial context, it is called HMRF, hidden marker random field. So there are a number of hidden variables, G's, and the Y's are or observations or emissions. And there are relationship between hidden variables in a spatially aware context. And when using the statistical models or computational models, we can infer the hidden stage as well as the observation when given the data. And in such a context, we can identify the cancer clones, tumor phylogeny, trajectory analysis, and so on. The second example is the spatial diffusion. Maybe we can think of the thermal diffusion, fluid dynamics diffusion, or mass diffusion. In such a sense, we can identify the distribution of drugs, distribution of metabolites, or the diffusion of cell-cell interaction or pathophysiology and so on. This type of diffusion should be understood in the spatially aware manner, and it can also be combined with ODE, ordinary differential equation, or PDE, partial differential equation. So when you are using the spatial diffusion, we are always thinking of the mathematical modeling of the system, and we can simulate this type of analysis. Here, the G-Fong is using governing equation like thermal diffusion, or like here, the blood diffusion of the part in a spatially aware manner. And the third type of example is the CNN, the convolutional neural network. Here, the input image may be the 3-channel image, like RGB image or grayscale image, or like monoscale images. And in such a case, we are using different types of kernels, and we can extract the local features of the images. And when we are constructing the convolutional neural network in a way of low-level features, mid-level features, and high-level features, and we are combining with that type of models with trainable classifier, we can identify, yeah, this type of image is cat, this type of image is dog, something like that. So even if the CNN is only focusing on the local features of the images, it was very successful, and maybe we should incorporate this type of models when we are dealing with morphological information, because in the spatial omics, there is always histological images or fluorescence images and so on. The fourth example is the graph data structure. So, for example, this type of analysis is called STGNNKS. This is identifying the spatial domains, and it is representing its spot as nodes and the spatial relationship between them as edges. And by using graph theory or adjacency metrics, Affiliation metrics, and so on, or even graph neural network, it is very popular in the spatial omics data. When we are using that type of approaches, we can identify the domains, spatial domains for spatial information. For example, here the G-Fong is also using the graph neural networks to simulate the spatial diffusion of blood or even fluid or thermal diffusion. And another example is emergent property. So maybe we can think of different window sizes and the biological metrics, or like biological variability, or the gene expression variability, high-risk gene expression, or something like that, according to different window sizes. And maybe the biological variables can be different according to the size of the window sizes. It is called a spatial emergent property. So, for example, this one is the example of iStar. iStar is using vision transformer to enhance the resolution of gene expression by using histological images. And here it is using two stages approaches, like the given images is divided into patches, and that each patch is subdivided into sub-patches. And the sub-patches identifying and extracting the features, local features, and the patches is somewhat global features. By doing so, this model can identify different types of spatial emergent properties. And also, as you know, Visual HD is also providing different bin sizes. Actually, it is known as providing 3 different bin sizes: 2 micrometer, 8 micrometer, and 60 micrometer. Each bin size, it's spatial differential. Spatial analysis with different bin size can result in different differentially expressed genes, or different targets, biomarkers, and so on. This is a type of emergent property as well. And also we can think of the large language models. And actually this is one of my papers. For example, we can think of the large language models that are taking the input of the spatial context of each cell. For example, a specific cell has its neighbor cells like epithelial or multiple fibroblast cells and T lymphocytes. In such a case, that spatial niche can be interpreted as fibroblast enriched environment, isn't it? That type of information can be input into large language models, and that type of features can be actually identifying the spatial niche, and it can be also visualized into Unmet visualization as follows. But I don't believe that the LLM is getting more easier, so you don't need to worry about it. For example, there's type of pre-made predefined functions that easily call the pre-made the large language models. Actually, this is so easy example, and maybe a lot of functions dealing with the large language models are getting more easier. So I think it's worth pursuing for you as well. For example, here I mentioned I inputted fibroblasts, macrophage, and neural. A lot of cell types are input into the large language models, and I visualized the embedding extracted from the input. For example, that type of input sentences can be translated into 384 dimension vectors by using this type of model. And actually, by using that high dimensional embedding vectors, we can visualize them into 2D projection by using Tiffany or UMAP. And as you can see right now, the fibroblast is surrounded by another fibroblast cells, like fibroblast cell subepithelial connective tissue prostate gland, something like that. So I think really the language models are understanding the context of input sentence. So I think it's worth pursuing in many contexts. Maybe I can explain this in the later slide. And from now on, I'd like to introduce unmet needs in spatial transcriptomics. So as you may be well aware of, there are 2 different modalities. The first one is a spot-based or sequencing-based spatial transcriptomics, and the other type is image-based spatial transcriptomics. There's kind of trade-off. For example, the spot-based one has low resolution and high gene coverage, while the image-based spatial transcriptomics is somewhat reverse to the counterpart. So here we can think of Morphscope, Genome, and this is the up-to-date information about the panels they provide. So Morphscope, developed by BGen, is providing something like 300 to 800 panel genes, and Genome is providing a few hundreds to even 5,000. Very recently, it is providing 5,000 in panels. But I do believe that this type of 5,000 in panels are not that useful in many contexts, because they are not providing the gene panels based on biological significance, but based on optical crowding or variability or something like that. That's the technical interpretation. So when we are using that panel design, maybe we should think of the custom panels for your own interest or your own experiment. So the panel design, the limited gene coverage would be the issue for gene panels. So There are multiple approaches to resolve that issue. The first one is to increase the number of genes from the image-based spatial transcriptomics. Here, IB refers to image-based spatial transcriptomics, and BB refers to barcode-based spatial transcriptomics. And here, as you can see, IB has a little bit smaller genes covered, and while the barcode-based one has a lot of maybe 20,000 genes are there. So I think when we are focusing on the intersected genes between them, we can make a model to expect the other genes from the intersected genes. For example, this model is somewhat predicting the other genes from the intersected gene, and this one is predicting this type of gene expression pattern to express barcode-based one. Please note that barcode-based one and image-based one has different distribution of gene expression. And when we are introducing the discriminator, we can even, maybe discriminator is somewhat discriminating between two, but our aim is to minimize their difference so that the discriminator is not well working. In such a sense, even the image-based spatial transcriptomic gene expression pattern, which has a different statistical distribution from barcode-based one, can guide us to infer from two other gene expression data. So by introducing that type of approaches, we can even eventually predict the intersected gene expression pattern to total gene coverage prediction. It's possible, but I think it's still ongoing. Maybe for example, there are a lot of biological variability and platform differences are there. So it's not that easy. I think it's ongoing research, and it's also a unmet need as well. So another approach is to effectively design the gene panels. There are two different methods I know. The first one is GPS-FISH, and the other one is SCGs. They are all published in good journals, genome biology. And this is my personal experience, my benchmarking research. Here, actually, let me tell you more context. So as I mentioned, the language models, right? So we can think of the cell type classification accuracy from panel size transcriptome. So actually, the cell type annotation is based on whole transcriptomics. However, if we resize that gene into panel size one, we can think of, we can test the accuracy, cell type prediction accuracy from the panel size transcriptome to predict the labels acquired from whole transcriptome. And in such a case, actually, not that many algorithms can do that, but large language models can do that. So I utilize that. That's my background information. There's a number of panel genes of candidates, random genes, highly variable genes, and highly expressing genes. And also, I'm utilizing the SCGs and GPS-FISH or cell type differentially expressed genes, or even the panel genes plus a manual curation. Here, the higher the value, the better. And as you can see, the SCGs has highest value, so that, okay, it's good, according to my previous experience. So I think I recommend many people to use SCGs for their own experiments when it comes to designing the panel. And also, the second problem I met is the sparsity. So the image-based spatial transcriptomics is problematic regarding the sparsity. So general single-cell RNA sequencing, UMAP embedding is somewhat clear. The clusters are clearly divided. However, when we are drawing the UMAP visualization of the image-based spatial transcriptomics, their clusters are not that separate, clearly separated, even if their histological or biological significance is a lot. So I think it's kind of a problem. This is very good example, and I think it's good. I mean, when the sparsity is higher, the cell type specific dot plot is not well drawn. I mean, it's okay. I mean, it's another. I mean, it's not good. I mean, so counterpart of that sparsity example. And also newly emerging data sets are there. So I met 3D spatial transcriptomic stuff. For example, morphish can acquire the information in a 3-dimensional way because it can also utilize the G dimension. And also multiple slices of spatial transcriptomics can give us the 3D spatial transcriptomics data. Also, very recently, a research group has pronounced that 3D spatial transcriptomics can be acquired experimentally by using tissue clearing approaches. So, I mean, the newly appearing data sets are there in spatial transcriptomics context. Also, the subcellular transcriptomics would be of interest for you. It's well known that RNA clusters are moving together in the subcellular compartments, and there are multiple algorithms to address that issue. For example, certain gene clusters are located near the periphery of the cell, while certain clusters are located near the nucleus or inside the nucleus. So, I mean, maybe it could be of interest to many of you guys. Also, cell segmentation is also unresolved issue. For example, this one is a PROSEC. Actually, there are a lot of algorithms: Bayesian, self-post, PROSEC, STARDIST. But for example, here the cell segmentation is not accurate, and even some approaches are using kind of expansion from nuclei, but it's not fair because some cells are having their nuclei in the one side of the cell, but that expansion algorithm is not considering that type of different nuclei positioning. And also, even if we many of scientists are trying to using digital pathology model, their pen marking is there, a lot of artifacts, a lot of poor quality data sets, but it's not easy to eliminate that type of poor quality data sets because I mean it requires human manpower, it requires time, it requires a lot of cost. It's not good, it's not automatic, it's not scalable. So I mean it's kind of issue. Also, morphotranscriptomic analysis should be there. There are a number of approaches to do that. For example, here, starfish, it is a starfish algorithm. It is considering the gene expression input and histological input. But as you know, there are different distributions in gene expression and histological images. So it's hard to harmonize them into one single embedding. So I mean, even if this algorithm is appeared, I think it's not widely used because it is not that well dealing with the both gene expression and histological images information. Also, resolution is issue. So when it comes to spatial transcriptomics, there are a lot of computer vision or digital pathology researches. It is using histological images. It is connecting the histological images with gene expression to predict certain score, to predict a certain chromosomal instability or certain cell type. It is widely used approaches over the world. But what about the resolution? I met several scanners. The scanners are characterized by MPP value, microns per pixel. Maybe the value range from 0.1 to 0.5. They're all different. And the models are maybe hard to find a certain specific values. For example, if a certain model is targeting higher value, I think it's not well working because many information are not that resoluted. I mean, it's dependent on low resolution image. It's not good. Or even a certain model is dependent on high resolution images. Maybe it will not be applicable to many data sets. So there's kind of trade-off. And I do believe that 0.5 would be a little bit consensus because this transpath is a kind of very popular vision transformer model, but even very recently, I found a paper from Google, which is developing a kind of digital pathology model. It is dependent on the 0.5 value. This is the paper about this algorithm. And then I think it's not easy. So even a lot of companies, a lot of institutions or research teams are making that type of digital pathology model, but just because of that resolution issue, it's not easy. And maybe even some other guys are trying to make a resolution agnostic digital pathology model. And also, I could name the other issues. For example, unstructured data in spatial transcriptomics. There are a number of different platforms in spatial transcriptomics. So it's not that easy to make a scalable approach to do that. And also, regarding the volume, regarding the resource, and regarding the speed, it's not easy to analyze the spatial transcriptomics, maybe 30 different samples, 100 different samples. It's not easy because of the resource. And also, there are a lot of fake information from the spatial transcriptome analysis, but we can tell the information from the noise. Even many bioinformaticians have suggested that the neuronal signature in cancer cells are not that accurate just because, just because of the gene interest, it's not fair to interpret it as a neuronal signature. But very recently, even nerve can be found in cancer microenvironment. So the neuronal signature makes sense. But who knows? Who can tell the information from the noise? Who can tell this is fair, this is right information, this is fake information? Who can tell that? I think it's unmet needs, and many biostatisticians are trying to understand that differences and maybe causal inference. And from now on, I'd like to briefly introduce the interactive visualization tools in spatial biology. So as you know, spatial biology is very special compared to the other former platforms like single-cell sequencing or bulk RNA sequencing because of the visualization. The decision makers are dependent on visual images. Just because we can see that images of gene expression, maybe it can be more credible. It can be more accurate, seemingly accurate. So I think even very good companies and institutions are trying to make their own visualization tools to make them cool, to make them very, very great. So I named a number of important visualization tools of interest. For example, NeuroGlancer is somewhat dealing with connectomics data, which is developed from the Google science team. Also, the Marscope team has provided the Marscope visualizer, and as you can see, there are a good functionalities here, and also Genome or cell by gene consensus has also provided different visualization tools. So, for example, here, maybe we see the NeuroGlancer can provide paired images, and actually it can move, the pointer can move at the same time. So there are a lot of button there, and maybe it's kind of 1 petabyte data opens, public data set. So they published 1 petabyte data. So I think maybe we can find a lot of serendipities from that data, and they provide their own data set with that visualization tools. So I think it's cool. Also, the Marscope visualizer, where Genome Explorer, where cell by gene data, they are providing their own functionalities, their user interfaces, and so on. So it's good, and I think even other teams are making their own tools. I met dozens of visualization tools. So maybe you can explore. Maybe you can share your own research with the visualization tools in your paper, in your conference, and so on. I just mentioned that because it's a hot trend nowadays. Okay, so my introductory material is over, and I'd like to introduce the code snippets I have made. I already ran some codes, and I'd like to introduce different topics, maybe beneficial to your own approaches. Spot deconvolution, cell type deconvolution, which can be absolutely independent of reference or partially dependent of reference and absolutely dependent on reference dataset. I think set type Q2 is partially dependent on reference, and cell dart is actually I utilize that algorithm a lot, but this is a lot dependent on the reference dataset. For example, there is another algorithm named STD-Combor, which is developed from Johns Hopkins research team, which is absolutely independent. Independent on reference data set. Also, distance analysis is very important compared to single-cell sequencing data. So I met a number of approaches in spatialomics data, but many other approaches are very similar in the single-cell RNA analysis context. It's not good. What's difference between spatialomics data and single-cell data? I think one of the key features is the distance analysis. So I would want to introduce this material to you guys. And also morphotranscriptomics analysis. Maybe we should combine the histological image when it comes to gene expression images. It should be, because there's a lot of information in histological images as well. And lastly, I want to mention cell segmentation using started. It's very easy to use. And maybe it's unmet need, and maybe a lot of image-based spatial transcriptomics downstream analysis is dependent on the performance of cell segmentation approaches. So I think I want you to make your own approaches for cell segmentation algorithms, because even the cell types annotation or cell allocations are not that accurate according to the cell segmentation results. So I want you to introduce how to design your own cell segmentation algorithms from the beginning. Okay, let's get started. So I already learned some installation part and loading some packages material, and I'm using the publicly available data sets from the SCSAP-TF package. And here 2,600 cells and 18,000 genes. And actually the data structure is something like that. OBS deals with the metadata for cells, and bar deals with the metadata for genes. So when we are looking at a data.obs, we can see a number of barcodes. Actually, it represents the cells. And here in tissue equals in tissue 1 means it is really on the tissue, but in tissue of 0 means it is outside of the tissue, so that it should be neglected. And array row, array column would be the spatial coordinates, and maybe some other metadata can be generated by your own approaches. For example, we can make you can make your own metadata right here. In such a case, you can see your own column from the OBS object. And also, as I mentioned to you, data variable information in the data and data. Here it is dealing with the metadata from regarding the gene. And here a lot of information, you can use that. Also, I'm also using UNS to make my own palette, color palettes. And also there's kind of embedding information as well, PCA or UMAP, something like that. And also OBSM would be there. I think it's good because it is storing embedding and spatial information, as well as image coordinates and so on. I'm not that using bar M and OBSP a lot, but a lot of keys are there in the end data. And here I'm using the brain data set here. So as you can see, this is coronal section of the mouse brain data, and a number of annotation is already provided. And here I want to mention spot deconvolution, which is also called cell type deconvolution. Cell type is good. It's good. I mean, it's already providing some reference data set. So we see we can insert the cell type and then we can see what models are there. And actually, it's getting more, I mean, evolved. You know what? I mean, when I used the cell type from the beginning, I met only 15 or 10 different reference data sets, but now it's providing more than 50 different data sets, reference data sets. So I'm using this mouse for brain data because our data set is somewhat adult mouse brain whole section. So it's good to use that. And actually, there's kind of a dependency issue in the cell typist when it is running in the under Google Colab. So even if I started with cell typist installation, I reinstall this one into our inferior version. It's not, I mean, it requires time. And as a result, we can identify that type of cell type annotation. And one of the key features regarding the cell type is the scalable approach. So when we are going to the official website of the cell typist, we can see automation or robust models, easy to use, a global reference. So I think the purpose of this model is to provide the approach to be used in multiple data sets, a lot of data sets, or scalable approach, and so on. And it's already it requires time. Also, maybe, okay, so that's the issue. That's the dependency issue I met frequently when I am working with Google Colab. So I already ran this algorithm under my own environment. It's so quick. I mean, if you are having a lot of resources, the typist is very quickly learned. But for example, it depends on the Python version. I think Google Colab is using higher Python version, like Python 3.11. It's a so higher version. In my case, my own environment only provides 3.8. But in such a case, I'm not using all of the possible references. Only 44 references are available in this specific environment. So it depends on your own environment. It depends on your packages. Even Google Colab is using higher Python version. It is not well working because a lot of packages are not that aligned with that higher Python version. It is a problem. And another algorithm I want to introduce is CERT-DART. CERT-DART is what I'm using, and it's kind of dependent. It has dependency on reference data set. So I already downloaded the reference data set from here. I'm using the gene expression omnibus archive a lot to approach. Wait a second. And this algorithm is using adversarial domain adaptation approach. Here, let me tell you this algorithm. So when there is a single cell RNA sequencing data, it is making a kind of pseudo spot by mixing the single cells. And it is comparing the real Visium spot against the pseudo spot. And the aim of this algorithm is to minimize the difference between two by using discriminator I have explained before. And maybe if that pseudo spots and real spots are so close, then by modifying the cell type proportion, then maybe that's it. that's the way of working. And I already downloaded the single-cell sequencing data. It requires exon count and metadata. So gene expression count and cell type label, that's all. That's a requirement. And it is, I'm just importing the packages, and I'm just pre-processing the single-cell sequencing data set as follows. And maybe mitochondria quality control, log transformation, normalization, and so on. And then I can meet type of single-cell sequencing embedding results. And maybe if I am using this type of function, I can acquire the differentially expressed genes according to cell type. And here, as you can see, a number of cell type marker genes are shown according to cell type in the format of table. And I can remit the number of markers, and this type of simple processing, and eventually the adversarial domain adaptation is working here. And I provided this code snippet in my own GitHub material, so you can also refer to my material as well. And as a result, you can see a number of different cell type proportion, visualization results. And when summing up all the cell type proportion, it should be 1 for each spot. Unfortunately, it wasn't functioning due to the version conflict with TensorFlow in Google Colab, so I will skip this part right now. And I want to jump into the distance analysis between clusters. I have seen many contexts that are using CKD tree or KD tree. So let me explain what is meaning about, and maybe I can encourage you to use this approach in your own approach when it comes to distance analysis between different variables. So here, for example, when there are 6 data points in the spatial context, we can think of, okay, the data point A can be a position in the middle of the data points. In such a case, A should be the root. And then we can think of different planes, and we can subdivide each half plane into 2 by finding the middle point of it longitudinally or horizontally. I mean, it should be taking turns. So we can find that type of tree that is really reflecting the spatial relationship between different data points. So even if there are a lot of data points, the tree architecture itself can provide a lot of fast approaches to find a specific nodes or specific relationship between distances. So in such a case, structure of a KD tree, constructing, the first step is the constructing a KD tree. And the second step is calculating the shortest distance using a KD tree. And maybe you can read them. For example, if a certain point is here, we can think, oh, it should be in close to B or close to D. But maybe as we have defined the division of the spaces, we can easily find this specific data point, the closest data points of specific point. So I'm using the cluster label from the A data. We see it takes, I think it should be stop. So I'm now using the cluster distance analysis. And for example, when we are exploring the Cortex-1 to Cortex-2 distance analysis, we can think of this type of analysis and visualization. Here, this plot visualizes the cumulative distribution function, and this plot visualizes the probability density function in the way of histogram. And when I am exploring Cortex-1 to Cortex-2, Cortex-3 or Cortex-4 and 5, there are maybe 5 choose 2, a lot of combinations are there. And I'm using that code snippet to identify the spatial relationship between different cortex cluster. And here, for example, cortex 1 is somewhat closer to cortex 4 compared to the other cortices. Let me see, is it true? I think cortex 1, blue, is really close to cortex 4 in a spatial distance, so it makes sense. And I think the CKD tree is very effective, even also in the context of image-based spatial transcriptomics. Please note that Visium spatial transcriptomics only provides thousands of spots, but image-based one can provide 1 million, even 1 million cells. There are a lot of cells. Maybe if we are using k-nearest neighbor or something like that, that requires n cube time complexity. It's not possible to run the image-based spatial transcriptomics by using k-nearest neighbor. It's not possible. But the KD tree, as it is using the tree architecture, the time complexity is a lot shorter than the k-nearest neighbor. So I have seen a lot of contexts that are using CKD tree when it comes to the distance analysis. I think it's good. I'm not sure. I couldn't find the other alternative for that. And I will jump into the morphotranscriptomic analysis. So actually, let's learn the first, and I will explain the other one. So let's see. So this is the algorithm provided in Spade. I already wrapped the hyperlink for that. So I can explain you what kinds of things are going on in this specific approach. So we can find a specific patch around a single spot, and that specific patch can be input into the convolutional neural network. There are a lot of predefined, pre-trained convolutional neural networks. I can name it, for example, VGG16, ResNet50, Inception V3, that are all made from the ImageNet challenges. And we can use that convolutional neural network to extract 512 dimensional vectors, and it can also be the metadata for that spot. And we can extract the principal components of that dimensional vectors, and that specific variable features could be related to the biological signals, but I mean, unsupervised way, right? So who knows? I mean, you should name it. Signatures are there. So I think it takes time. So I mean, I want to skip that, but I want to show you a part of a few points of that. For example, it requires a TensorFlow, and actually the dependency is not high compared to the other AI models. And actually, you can easily bring that trained models, and you can use. I mean, maybe this one is connecting the spot to image subpatches. So let's see. I think it takes time.
Colabs are great because we can share our code, but unfortunately it never saves what we run. So that's why we always have to install.
Yeah, it's kind of dependency issue is prevalent.
So you get the code and you can run it on your own and save it nicely, but unfortunately, every time you run it online, like every time you run it online, you'll have to reinstall everything.
I was thinking one of the aspects of spatial that I mean, of course, DNA is different, DNA mutation is different, but when it comes to expression, protein or RNA is at that time, if it expressed, it makes it. So that's one of the problems that most scientists run into. They give you the FFP your analysis on this particular time. Like I'm taking a picture of you right now. You're not sitting there 24 hours a day every day.
Yeah, exactly. Yeah.
So that's the problem. And that's why I think like either you liquid biopsy or or needle biopsy of a tumor, a different area.
Yeah.
Could be the like over time could be the best way to really understand like the if you're doing phylogenic and all of that. You cannot really do it on gene expression.
Yeah.
You cannot capture it.
Yeah.
That's one of the problem I see. It's the temporal aspect.
That makes sense. Yeah. Because it is destructive still, right? As you're saying, you need some sort of biopsy if you're in the, so you can't.
You can just based on like months and months of analysis
Right.
on one spent time.
Right. Yeah. You have to.
Yeah, and then your conclusion based on that one snapshot. That's just
Not very
there's
thorough.
no common sense of that.
Yeah.
I was just thinking.
Yeah, that's a really interesting point. Yeah. I wonder, I mean, like, yeah, you could make models, right? Like, and do those across time, but as you're saying, in
Yeah,
patients.
like organoid models.
Yeah.
If you do organoid models where you can do spatial omics on those across different times. Yeah. But I think the best thing is just to get biopsy and look at biopsy patients.
Oh, yeah, that's a good idea too. Yeah. Collecting enough patient biopsies, would allow you to make some sort of temporal ordering.
Because patients, what are the chances that you get 19 patients? These are patients that are many different treatments and do many different kind of treatments that cause different expression.
Yep.
So when you get a frozen tissue or FFPE sample, you never knew what's their background, what kind of treatment did they do? Did they undergone that could destroy certain expression of RNA and protein or if we
Absolutely.
You
Your
see all of-
signals,
like all of that has to be put into consideration when you make the story or phylogenetic trees about a particular.
The biology is such a complex
It's
system
so complicated.
that if you just look at one time point in one area, it doesn't.
Not enough.
Yeah.
And then if you base on your conclusion just on that, it's just not right.
Yeah, that's very interesting. Yeah, you're right. So these are cool tools that allow you to see more information. But the problem of lack of data to begin with is not resolved. Yeah.
So you can spend time and trying to use the tool on one snapshot. It's a waste of time. I think.
Yeah. Unless you can actually collect enough samples and analyze all the
Enough
samples.
data and know the background data. Where is it coming? What kind of treatment? They only genomic DNA data.
Oh, I don't know. I was just gonna write this down.
Oh, okay, right. They would just only do the genomic data, DNA changes within patients that has undergone different treatments. And just do that and provide a whole atlas. If this certain patient, certain disease, but with this treatment, this is the DNA mutation.
Mhm.
This is the difference, but that's the only thing they could do with DNA mutation. It would be amazing to look at
High throughput, yeah.
high throughput using spatial
Yeah,
of protein and RNA by observing the patients over time.
Mhm.
And we can really do it through liquid biopsy
Mhm.
and needle biopsy in different area over time.
Does a liquid biopsy maintain the spatial organization?
You can,
Yeah.
because that's when you get supernaturally cells, for example,
Mhm.
cancer.
Mhm.
If it has to do with cancer, of course you can do that. But certain kind of disease that could be
I see.
migrating in. You can put it on size. And also, tumor, you can also have a tissue.
Over time.
After certain steps.
Yeah, let me give wrap up this session. So I just want to mention, actually there's kind of dependency issues right now. Actually, I recovered the version originally, and also I want to introduce this type of classifier. It is published in Nature, and I think it's even if the code snippet is so simple, I mean, you know, the scikit-learn has provided a lot of classifier. However, I think it's well working. Even the tabular data can be processed using foundational model. And I just want to introduce this type of auto in your own approach. There are some problems like number of features are limited, the number of data sets are limited, but even if so, this kind of tabular data foundational model. So maybe you can use that. I just want to introduce, and maybe you can incorporate this type of model in your own research. That's all. Actually, the start is not working as well because the TensorFlow version conflict issue, but it's fine. I mean, you know, you can use your own data set and algorithm in your own environment, as I've shown to you. So that's all. Thank you for coming here, and let's call it a day.